Hello, my name is Tatiana Joaquin, and in this presentation, I'm going to talk about the enzyme lactase. I will provide an overview of the enzyme, including its physical structure, the reaction it catalyzes, and reporting on some reaction kinetics. Lactase is found in the brush border of the small intestine of many mammals. It is a digestive enzyme. Lactase is also found in the lactose degradation pathway. It is the enzyme which catalyzes the reaction. We will discuss this more momentarily. Finally, we find lactase in lactose-free milk products. The added lactase breaks down the lactose present in milk for those who cannot digest lactose. The main reaction which lactase catalyzes is lactose degradation, also called lactose hydrolysis. This mechanism is part of a metabolic pathway to supply energy to the body. If you look at the structure of lactose, you can see that it is made up of two sugars, glucose and galactose. The two sugars are connected via a glycosidic linkage, which is a type of covalent bond. The glycosidic linkage is where the two monosaccharides bridge together with the oxygen. The function of the lactase enzyme is to target the beta-galactoside and break the glycosidic linkage. When the glycosidic linkage is broken, lactose splits into its monosaccharide subunits. Lactose is the main sugar found in milk products. When in the infancy stage of life and relying on milk as the primary source of nutrients, lactase production is high. As we age, lactase production decreases. Without lactase, we cannot digest lactose, which can lead to lactose intolerance. Lactose intolerance comes with many uncomfortable side effects, including bloating, abdominal cramping, nausea, and more. Around 65% of the world population is lactose intolerant, yet many of us continue to consume milk products. That is why many lactase-enriched, lactose-free milk products are in supermarkets now, so people who are lactose intolerant can still enjoy their ice cream. What we are looking at is the RCSB Protein Data Bank website. This website allows you to look up the structures of many proteins. The structure for lactase that I liked was the one shown here. It is actually of the enzyme substrate complex of lactase bound to lactose. An enzyme substrate complex is the temporary molecule that forms when a substrate and enzyme come in contact. Sometimes the term beta-galactosidase is used instead of lactase. This is because lactase belongs to the beta-galactosidase family of enzymes. This family of enzymes works by targeting disaccharides and breaking glycosidic bonds. The 4-character PBD ID for this version of lactase is 1JYN. Directly above the enzyme, there is a series of letters. This is the primary amino acid sequence which gives rise to the structure and function of the enzyme. Looking at the enzyme, there are several places where a set of two connected spheres are present. These are the lactose substrates. When I click on the button to hide carbohydrates, the lactose disappears. This is because lactose is a disaccharide, which is a sugar. And what are sugars? Carbohydrates. If I click on one of the lactose molecules, we can see the active site of the enzyme. The active site region of the enzyme is where the substrate binds. Around the active site are magnesium and sodium ions to help with binding. The lactose is bound by many hydrogen bonds into the active site region. Now we are looking at the Swiss PBD viewer. On the right side of the screen are all the amino acids that make up the primary sequence of the enzyme structure. Some of the boxes are colored red or yellow, showing which amino acids contribute to alpha helices or beta sheets. On the left of the screen is the complete enzyme, showing its tertiary structure. Within this structure, we can see alpha helices and beta sheets, which make up the secondary structure of the enzyme. Alpha helices are colored red and beta sheets are colored yellow. This coloring scheme matches with the amino acids box. There are two common ways to measure lactase activity. We can measure glucose levels in a solution over a period of time using glucose test strips. The other option is to use an ONPG analog instead of lactose as the substrate. When ONPG is catalyzed, it is split into galactose and orthonitrophenol, ONP. The orthonitrophenol has a yellow color. Colorimetric and spectrophotometric analysis can be used to measure enzymatic rate based on the appearance of ONP. The steps to measure lactase activity are fairly straightforward. Begin by setting the wavelength on the spectrophotometer to 420 nanometers, which is the absorbance for yellow solutions. Next, blank the spectrophotometer with the phosphate buffer solution. Then add a specified concentration of phosphate buffer to the cuvette, followed by the ONPG substrate. Next, add the lactase enzyme into the cuvette, place a piece of parafilm on top of the cuvette to cap, then invert to mix. Ensure to immediately place the cuvette into the spectrophotometer and begin timing the reaction. Finally, measure the absorbance of the solution at a determined time. The reaction rate is the amount of substrate converted into product over a specified amount of time. The rate at which the intensity of the yellow color increases allows us to calculate the enzymatic activity of lactase. By measuring the absorbance at different time points using the spectrophotometer, we can construct a standard curve diagram. From the standard curve, the initial rate, V0, of the reaction can be calculated. From the graph of V0 versus ONPG concentration, Km can be estimated. We can also construct a Lineweaver-Burke plot by plotting 1 over V0 on the y-axis against 1 over substrate concentration on the x-axis. Vmax, Km, and Kcat can be determined from the constructed plots. The experiment should be repeated numerous times to ensure accurate data collection and kinetic values. Km is the affinity of the substrate lactose to the active site of the enzyme lactase. A typical Km value in the bacterium E. coli is 0.12 millimolar. Kcat, also known as the turnover number, describes the number of lactose molecules that are turned into products, in this case being glucose and galactose, per unit time by a single enzyme. A typical Kcat value in the bacterium E. coli is 620, 1 over S. Lactose, sucrose, and maltose are all disaccharides. Looking at the structures, they all look similar. So why is it that lactase will bind to lactose to catalyze lactose degradation, but will not break down sucrose or maltose? Look at the box containing the disaccharides. In it are lactose, sucrose, and maltose. Each disaccharide listed is made up of 2 monosaccharide subunits. The subunit combination for each disaccharide differs. On the lactase enzyme is a specific region called the active site, which will fit the shape of one of these disaccharides. Which one will fit to be the substrate? I hope you said lactose for 2 reasons. The first reason is that the shape of lactose is the only one of the 3 which will fit into the active site on the enzyme lactase. The second reason is that sucrose and maltose will not be broken down because lactase is a beta-galactosidase enzyme, which breaks a glycosidic linkage at a beta-galactoside. A beta-galactoside is a galactoside where the glycosidic bond is above the galactose molecule. Lactose is the only one of the 3 disaccharides above which contains a galactose molecule, and thus a beta-galactoside. In the next video, I will provide a further explanation on the mechanism of lactose degradation, including the inhibitors. I will also provide an overview on the catalytic strategy of lactase. Please like this video if you found it helpful and comment with any questions. Thank you for watching, and I hope you learned something.